For kind of introduction, it is very we heard a very important introductory lecture from Dr. Ridke. He mentioned residual cholesterol risk as well as residual inflammatory risk. I propose the PCSK9 has two different mechanisms to induce the atherosclerosis. Yes, obese inflamed fat is associated with cardiometabolic disease such as obesity and cardiovascular disease and diabetes as well as cancer. What happens in this abdominal obese fat is shown here. There are two fundamental physiology undergoing during the transition from lean to fat, abdominal fat. The first one is hypertrophy of adipocyte. So the second is infiltration of macrophages. These two happening induces secretion of many cytokines mentioned before, causing atherosclerosis and insulin resistance on muscle and liver. As you know, adipose tissue is not only energy reservoir organ, but as well as endocrine organs secreting large amount of cytokines, including resistin. That is my topic since 15 years ago. We first report resistin is causal factor of atherosclerosis. Resistin, known as adipokine, is a key player to modulate monocytes, leading to progression of atherosclerosis. Here's the mechanism. There are four key players of atherosclerosis: mono, macrophage, and endothelial cell, and smooth muscle cells. When they were exposed to resistin, the change happened in all these four key players: induction of VCAM1 and integrins. The result is shown here: enhanced adhesion, invasion, retention, and survival, leading to progression of atherosclerosis. When I published this paper, I tried to block the resistin signal, but at that time, there's no known receptor for resistin. Thus, I tried to clone the receptor for human resistin. At the 3 years endeavor, we finally cloned the human resistin receptor, that is adenylyl cyclase associated protein 1, CAB1. RISC-CAB axis mediates inflammatory actions of human monocytes. In other words, resistin can bind CAB1, leading to induction of cycle AMP, pKa, NF-kappa B, and many cytokines in monomacrophages in human. The signaling pathway is shown here: RISC-CAB, adenylyl cyclase, cycle AMP, pKa, and NF-kappa B, and many cytokines, including interleukin 6. So for the last 10 minutes, our lab focused on the big map, how RISC-CAB axis induces many disease conditions. First, in the obese abdominal fat, we have many infiltrating macrophages that express high level of resistin. Interesting findings: CAB is ubiquitously expressed in all human body: liver, artery, intestine, muscle, even cancer cells. Thus, interaction of resistin from monocytes and CAB in many parenchymal cells lead to the disease. We generate TALEN-mediated transgenic CAB knockout mice. Homo-knockout mice were lethal at the embryo day 15. Thus, we were able to analyze the phenotype of hetero-knockout mice. The first phenotype is reduction of fat inflammation under high-fat diet. Here's wild type, animal, chow diet, high-fat diet. There's huge infiltration of brown macrophages. In other words, it turns as fat, inflamed fat, remarkably reduced by CAB hetero knockdown. Pro-inflammatory gene expression in this fat tissue is also decreased by CAB hetero knockdown. Thus, we now summarize the paper that RISC-CAB axis is a very important mechanism to explain the metabolic syndrome. The second phenotype is reduction of hepatic steatosis under high-fat diet in hetero-knockdown mouse. Here's wild type, fatty liver, like foie gras, significantly reduced in CAP hetero-knockdown mice. Lipogenic gene, gluconeogenesis genes are turned down in hetero-knockdown mice. We also summarized the paper to decipher risk of X is very important pathophysiology to induce NASH, hepatic steatosis, and liver cirrhosis. The third phenotype is also very interesting, reduction of LDLC by 15% in hetero-knockdown mice. There are two key players to control LDLC, receptor and PC9. Thus, we checked hepatic expression of receptor and PC9 in these hetero-knockdown mice. Here's wild-type mouse under chow diet and high-fat diet, receptor, CAP, PC9. Hetero-knockdown mice, CAP is physiologically knocked down by about 30 or 50%. PC9 does not change, but receptor are accumulated. It's very interesting finding. I guess there may be some defect in the interaction between PC9 and receptor, leading to accumulation of receptor and decrease of LDLC in blood. Thus, we started this big project since 5 years ago, and we confirmed CAP is binding partner of PC9 and is required for the degradation of receptor by PC9. Here is a hepatocyte surface is enriched with receptor that uptake circulating LDLC from blood, and cholesterol is used to synthesis of bile acid. Receptors are recycled. In the absence of PC9, there are a lot of receptors on the surface, thus decrease of LDLC in blood. But PC9 is also synthesized in the same way like LDL receptor, the same transcription regulator of SRABP2. When it binds to receptor, receptor cannot be recycled and degraded. Thus, receptor number on the hepatocyte are few, leading to increase of LDLC in blood. Thus, they develop PC9 inhibitors, monoclonal antibody, to block the binding between PC9 and receptor, leading to receptor recycling and LDLC decrease in blood. Here's binding site of receptor and PC9. EGF precursor homology domain of receptor bind to catalytic domain of PC9. There are 3 domains in PC9. But since about 5 years ago, at that time, nobody knows the mechanism of endocytosis and the lysosomal degradation of receptor after binding with PC9. We found a very interesting phenotype of CAP hetero-knockdown mice, accumulation of receptor without any change of PC9. Thus, I guess there may be some defect in interaction of the PC9 and receptor. We found a very interesting paper. Here is the PC9 structure. Cysteine rich domain looks like resistin. That is the starting point of our project. We already published structure of resistin trimer CAP binding complex shown here under the collaboration with Dr. Sang-Ak Lee on the panel. And we found the paper resistin is very similar to PC9. C-terminal structure of resistin is very similar to cysteine-rich domain of PC9 on profile, on en face. We propose this protein docking model, thus we expect such kind of protein docking model between PC9 and CAP. we established 3D modeling of PC9 cap docking simulation like this, and we confirm that PC9 directly binds to cap through cysteine rich and SH3 domain, like this. At the receptor bind to catalytic domain of PC9, the exposed free cysteine rich domain bind cap through G3 domain. Such evidence is proved by 4 different methods. First, immunoprecipitation. Second, far western blot. Third, by molecular fluorescence complementation assay. 4, surface plasmon resonance assay. And furthermore, we try to map the binding motif using a series of deletion mutant cap. G3 domain of cap is the motif for binding with PC9. Conversely, using a deletion mutant PC9, we confirmed CLD of PC9 is indeed the binding partner of cap. Thus, this is the answer of our experiment. Such a notion is well realized in human real genetics. We confirmed the loss of function mutants of PC9 such as 668, 670 could not bind with CAP1. This mutation is reported in human. There are several mutations of PC9 gene in human, such as like this: bad mutation, gain of function; good mutation, loss of function, encompassing exon 1, 2, 3, 8, and 9, 12. This is prodomain, catalytic domain, and cysteine rich domain. This is very important motive to bind CAP. Thus, we focus on 8 gene mutations, 2 bad mutations, 6 good mutations. And we prepared from a laborious laboratory work, we prepared all 8 mutant proteins of PC9, which is reported in human. Among 8 mutants, these 2 mutants showed defective interaction with CAP1. Here's the evidence. Bad mutants and good 6 mutants. Bad mutants degrade receptor. Gain of function mutants. Good mutants, loss of function mutants, enriched receptor. Among them, black is nonsense mutation, no PC9, really a longevity phenotype. And there are green, 4 green and good mutants. Among them, these 2 mutants on cysteine rich domain cannot bind with CAP. Thus, our notion is well revealed, corroborated in real human gene. Here is these 2 mutants cannot bind with CAP. That's the reason why there is a loss of function and decrease LDLC in blood. What's the mechanism for internalization of receptor PC9 CAP complex through caveoli or clathrin? There are 2 main endocytosis mechanisms, clathrin and caveoli dependent endocytosis. When we applied caveolin 1 siRNA, PC9-induced receptor degradation was disturbed. Caveolin is very important. When we block clathrin, there is no change of receptor degradation. Clathrin is not important. Here is immunofluorescence staining. In control conditions, receptors are degraded with lysosomal activation. Such if happening does not change even we block clathrin. Clathrin is not important. When we block CAP or caveolin, receptors are not degraded even under treatment of PC9. Here's another evidence. In control conditions, PC9 degrade receptor, which is not affected by clathrin siRNA, but receptors are not degraded with CAP or caveolin 1 siRNA. Thus, CAP caveolin is very important protein to degrade receptor by PC9. Here is the actual quadruple complex domain. Ejectricus homologous domain of receptor bind to the catalytic domain of PC9. CLD bind C3. AC bind domain contact with caveolin. This quadruple complex internalized through caveosome, leading to lysosomal degradation. In this process, CAB is required for PC9-mediated receptor degradation, leading to increase of LDLC. If CAB is not there, even a lot of PC9 cannot degrade the receptor. PC9 is non-functional without CAB. Here's a PC9 treatment induced receptor degradation, which is blocked by CAB knockdown. Here, when we applied wild CAB, PC9 degrade receptor, but AC domain deletion mutant, C3 binding domain mutant cannot degrade receptor. These two domains are important, suggesting this model. Lysosomal degradation is important. When we apply the blocker of lysosomal degradation, receptors are not degraded. Proteasome inhibitor, autophagosome inhibitor does not have any impact on the degradation of receptor by PC9. Here's another evidence. Here, control conditions, receptor degraded. CAB knockdown, receptor maintained. Early endosome is not changed. In this case, degraded. In this case, recycled. Here, lysosome is activated, degraded. Lysosome is not activated, so recycled, enriched with a receptor on the surface of hepatocyte in the presence of CAB siRNA. Thus, this is summary. In the absence of PC9, receptor recycled. But in the presence of PC9, it may be recycled or it may be degraded. The decision maker is CAB. If CAB is there, it undergoes caveosome internalization degraded. Without CAB, even in the presence of PC9, receptor is not degraded. The other important thing is when LDL cholesterol level is decreased, there is the inflammatory risk. PC9 has also a player to induce the inflammation independent of receptor degradation. There are several suggestions the pro-inflammatory action of PC9 based on experimental and clinical evidences. Gain of function PC9 mutation increase such kind of cytokines. Until now, there are many PC9 resources, not only from hepatocyte, but also from monomacrophages, endothelial cells, and smooth muscle cells. These PC9 in plaque may induce expression of scavenger receptor on the surface of monomacrophages, leading to formation of foam cell. Thus, PC9 has some pathological role to progress of atherosclerosis. Until 5 years ago, there are some suggestions, the mechanism of PC9 to activate macrophages. PC9 can increase toll-like receptor 4, leading to NF-kappa B activation, or PC9 obliquely locks expression, enhancing the uptake of OX-LDL, leading to NF-kappa B activation. But it is unclear. For the recent 3 years, we fully deciphered how PC9 induces inflammation independently of LDL receptor. We prepared macrophages from LDL knockout mice, LDL receptor knockout. When we treat PC9, monocytes can be activated, like wild-type animal. Does PC9 induce NF-kappa B, but LDL receptor is not necessary. We also introduce AAV PC9 gene to LDL receptor knockout mice. Here, we can induce atherosclerosis independently of LDL receptor. When we harvest this carotid artery and check the cytokines, huge amount of interleukin-1, 6, TNF-alpha induced by PC9, even in the absence of LDL receptor. For the last 3 years, we did an extensive experiment and established such kind of whole map, how PC9 induce inflammation independently of LDL receptor. PC9 can bind CAP, which may influence the binding of PC9 to TOR4. This is main interaction. This is mild interaction. The downstream signal is adenocyclase, cyclic AMP, pKa, PI3 kinase AKT, and kappa B. PI3 kinase can turn on the adhesion molecules, and if kappa B turning on many cytokines. AKT can induce translocation of SIDBP2 to nucleus, leading to translation of PC9. This is very important self-augmentation pathway. PC9 induces PC9, as well as upregulation of TOR4 receptor and LOX1. The first, PC9 colocalized with carbon on macrophagy. PC9 treatment induced change of CAB localization to the membrane. Direct evidence, direct interaction between PC9 and CAB on the surface of macrophagy. Direct interaction between PC9 and CAB with Blitz experiment on the surface of macrophagy monocytes. Direct interaction of PC9 CAB in dual link experiment is shown here. Red dots suggest the colocalization of PC9 and CAB, which is not affected by TOR4 blocker, LOX1 blocker. But conversely, interaction with PC9 TOR4 receptor is dependent on CAB1. PC9 TOR4 interact, but which is significantly retarded in the absence of CAB. Thus, this is main binding partner. This binding, presence of this can facilitate the binding between PC9 and TOR4. This interaction is negligible. Then PC9 turns on NF-kappa B like this, and macrophage as well as EC. Then downstream pro-inflammatory genes, IL-6, are turned on by PC9 on macrophages. These molecules on macrophage and EC, VCAM1, ICAM1 on EC, integrating alpha-4, beta-1 on macrophages, thus enhancing interaction between two cell types by PC9. Such kind of kappa B cytokine activation is all dependent on CAB. When we knock down CAB, such kind of induction obliterated. But when we knock down TOR4, it's very weak effect. This main mechanism is PC9 CAB axis to induce NF-kappa B or many cytokines. Furthermore, PC9 induced scavenger receptor LOX1, scavenger receptor 1, CD36 on TOR4 macrophage, all dependent on CAB. Such a scavenger receptor is activated by PC9, blocked by CAB. Uptake of ox-LDL is induced by PC9, which is also dependent on CAB. Ox-LDL is uptaken by PC9-stimulated macrophages, but which is blocked by CAB, like this. Potent positive feedback loop is found. PC9 induces PC9 secretion from macrophages. Here, many bad materials induce PC9 secretion from monocytes, but PC9 is the strongest stimulant PCSK9 secretion from monocytes. But this pathway is also dependent on CAP. SREBP is turned on by PCSK9, leading to PCSK9 synthesis, which is blocked by CAP. In vivo relevance, yes, PCSK9 or low shear stress-induced vascular inflammation, atherosclerosis, is all dependent on CAP. Here's one evidence. Wild-type animal, PCSK9 injection induced atherosclerosis, which is obliterated in the CAP hetero-knockout mice. Another evidence, low shear stress itself induces atherosclerosis like this. But in CAP hetero-knockout mice, even low shear stress does not induce plaque because there's no infiltration macrophages. RISC-CAP-X is very important. PCSK9X is very important to induce macrophage activation. But macrophage is not activated. Even in the presence of low shear stress, no plaque, no cytokines. Thus, CAB is really pivotal receptor to induce, to mediate the bad actions of PCSK9. Thus, once again, CAB is pivotal molecule in inflammation and metabolic syndrome. It plays a role, a cholesterol risk. It also plays a role, residual inflammatory risk. But avalokumab does not have any impact on anti-inflammatory action because they block the receptor, a binding between either receptor and PCSK9. Even in the presence of avalokumab, there is still interaction between PCSK9 and CAP, leading to inflammation. Thus, to capture both cholesterol risk and inflammatory risk, we have to develop blocker against PCSK9 CAP binding. In our experiment, FC-CAP1 is very powerful to block cholesterol synthesis, to induce lowering LDLC, but also to block inflammation. Thus, such kind of blocker may really realize the control of cholesterol risk as well as inflammatory risk. Thank you very much for your attention.